Here is a comprehensive guide for simulating RNA folding sequences using quantum computing and Hamiltonian algorithms. Introduction. The process of RNA folding, where the RNA molecule takes on its functional shape, is crucial for understanding its biological function. Traditional computational methods for predicting RNA folding, such as dynamic programming, stochastic sampling, and genetic algorithms, have limitations in accurately predicting tertiary structures. The use of quantum computing and Hamiltonian algorithms offers a more efficient and accurate approach to simulate RNA folding. Step 1. Understanding Hamiltonian algorithms in RNA folding. Hamiltonian algorithms, also known as energy-based algorithms, use mathematical models to simulate the energy landscape of RNA folding. This allows for the prediction of the minimum energy conformation of the RNA molecule. Different algorithms can be employed for this process. 1. Dynamic programming, DP. DP algorithms, such as the Zucker algorithm, are efficient for predicting the secondary structure of RNA by minimizing free energy. They are deterministic and guarantee finding the optimal solution within their model, but can be limited by the simplifications of the energy model and may not always predict the correct tertiary structure. 2. Stochastic sampling. Algorithms like the Monte Carlo method sample the conformation space randomly, which allows them to explore a wider range of possible structures. However, they may require a large number of samples to find the global minimum and can be computationally expensive. 3. Genetic algorithms, GAS. GAS simulate natural selection processes to explore conformation space. They are good at searching large spaces, but can get stuck in local minima and may need careful tuning of parameters. Step 2. Combining algorithms for comprehensive prediction. Combining these algorithms can leverage their strengths and mitigate their weaknesses. Use DP to quickly find a local minimum or to understand the secondary structure landscape. Apply stochastic methods, like simulated annealing, to explore the folding space more globally and escape local minima identified by DP. Run GAS in parallel to explore multiple regions of the search space simultaneously. Integrate structures into a joint probability model to estimate the uncertainty in predictions and identify the most probable tertiary structures. Step 3. Sequential employment of algorithms. Employing algorithms sequentially can lead to efficient exploration. Start with a stochastic global search to sample the conformation space broadly. Use dynamic programming to refine promising regions identified by the global search. Apply machine learning techniques to analyze the diverse predictions and identify consensus structures, or to learn from known RNA structures to improve predictions. Step 4. Setting up the computational environment. 1. Software installation. Install a quantum simulator or access a quantum computing platform like IBM Quantum Experience, Rigetti Forest, or Quantum Azure. Install necessary software for RNA folding prediction, such as Rosetta RNA or RNAfold. Set up a development environment for programming and running quantum algorithms. 2. Data management. Set up a data management system to handle large amounts of data generated during the simulation. Use a cloud-based storage solution for easy access and collaboration with other researchers. 3. Visualization. Utilize software such as PyMol or VMD for 2D and 3D visualization of RNA structures. Develop scripts to automatically capture folding sequences and generate visualizations. 4. Parallelization. Utilize parallel computing techniques to distribute the simulation tasks across multiple computational nodes. Use a job scheduler to efficiently manage and allocate resources for the simulation. Conclusion. By utilizing quantum computing and Hamiltonian algorithms in simulating RNA folding, we can overcome the limitations of traditional methods and accurately predict tertiary structures. By strategically integrating different algorithms and employing them sequentially, we can enhance the comprehensive prediction of RNA folding. Setting up a proper computational environment, managing data, and visualizing results are crucial for a successful simulation. Parallelization of tasks is also essential for efficient and thorough simulations.